Greetings all. In the Level 1 video for cellular respiration, we talked about the balanced chemical equation for the oxidation of glucose. Here, for the Level 2 video, we're going to briefly discuss the different metabolic pathways that together make up cellular respiration in a eukaryotic cell. Glycolysis, which happens in the cytosol of almost every living cell, is going to chop the glucose molecule in half, making 2 molecules of pyruvate acid. Or, really, at physiological pH in humans, 2 pyruvate ions. If we count the atoms on the right-hand side of the equation, we have a total of 6 carbon atoms, 8 hydrogen atoms, and 6 oxygen atoms. On the left, glucose has 12 hydrogen atoms. We still have all 6 carbon atoms, so we haven't turned any carbon into carbon dioxide yet, but we lost 4 hydrogen atoms. Where did those 4 hydrogen atoms go? Our old friend NAD+ takes care of them as 2 pairs of high-energy electrons. For now, let's treat glycolysis as a magic box and look at it like this. We need to spend 2 molecules of ATP in order to get glycolysis started in what we're going to call the investment phase of glycolysis, but we're going to make 4 molecules of ATP before we're done with glycolysis. We might also be able to make some ATP from the NADH plus H+, if we can get these high-energy electrons to the electron transport chain later on. That's a profit of at least 2 ATPs per glucose molecule, which actually isn't all that bad. If you're an anaerobic organism, these 2 ATPs are all you ever get from 1 molecule of glucose. We have 2 pyruvate ions coming out of glucose, so everything from this point on happens twice per glucose molecule. The pyruvate dehydrogenase complex takes the pyruvate ion coming out of glycolysis and turns it into acetyl coenzyme A, plus our first molecule of carbon dioxide. Of the 3 carbons in pyruvate, we just lost one of them as carbon dioxide, and the other 2 carbons are the acetyl group of acetyl-CoA. We also squeeze out some high-energy electrons here too, and put them onto NAD+ to make NADH plus H+, which is where we get the dehydrogenase part of the name of pyruvate dehydrogenase complex. Next is the citric acid cycle, and probably the best thing about the citric acid cycle is how very many names it has. You can call it the citric acid cycle, or you can call it the citrate cycle, since at physiological pH what we actually have is the citrate ion, not the citric acid molecule. Or you can call it the tricarboxylic acid cycle, which makes sense when we look at the structure of citric acid here in a little while because it has 3 carboxylic acid groups. Or you can call it the TCA cycle when you don't want to have to spell out tricarboxylic acid and make sure you've spelled it right. Or you can call it the Krebs cycle, which is what old people like me were used to calling it. Or you can call it the St-Georgi-Krebs cycle, which I very much doubt I'm pronouncing correctly. These are both because scientists love to name things after dead people, in this case Hans Krebs and Albert St-Georgi. The citric acid cycle, which happens inside the mitochondrial matrix just like the pyruvate dehydrogenase complex, is going to finish making our carbon dioxide molecules and, along the way, is going to make high-energy electrons, which then get sent into the electron transport chain. The citric acid cycle is also going to make 1 molecule of GTP per cycle, which has the same sort of high-energy phosphoanhydride bond that we have in ATP, so the energy is the same. The citric acid cycle is going to take the 2 carbon group off the end of acetyl coenzyme A and stick it onto oxaloacetic acid, or in real life, the oxaloacetate ion, again, physiological pH, ionized, blah blah blah. Oxaloacetic acid is a 4-carbon thing, so when we take the 2 carbons from acetyl coenzyme A, we get citric acid, again, really the citrate ion. Citric acid is a 6-carbon thing. When we turn the crank on the citric acid cycle, we start with oxaloacetate, and 9 steps later, we end up with oxaloacetate again, which is why it's a cycle. Remember, we turn the crank twice per glucose molecule, so here are the last 4 molecules of carbon dioxide, which accounts for all of the carbon atoms that we started with in the glucose molecule. We also get 2 molecules of GTP and a whole lot of high-energy electrons. Most of the high-energy electrons are carried away on our old friend NAD+, but we also get to introduce another redox cofactor, flavin adenine dinucleotide, or FAD, which carries away another pair of high-energy electrons on FADH2. These are, functionally at least, not quite as high energy as the high energy electrons carried by NADH plus H+. Finally, the electron transport chain is going to take all of the high energy electrons we've made so far, minus any that we lost or spent along the way, and use the potential energy in these high energy electrons to create a hydrogen ion gradient across the inner mitochondrial membrane. The who with what now? Don't worry, we'll look more at the details of that in a level 3 video later on. FADH2 drops off their not quite as high energy electrons on the second step of the electron transport chain, while each NADH plus H+ drops off their high energy electrons on the first step. Every time a pair of high energy electrons drops down a step, they become a pair of slightly less high energy electrons, and the potential energy squeezed out of the high energy electrons is used to pump a hydrogen ion across the inner mitochondrial membrane. We're trading the potential energy of high energy electrons for the potential energy of a hydrogen ion concentration gradient. Later on, we're going to use the potential energy of the hydrogen ion concentration gradient to make ATP. 1 ATP molecule for each hydrogen ion that got pumped across the inner mitochondrial membrane. When the electrons get to the bottom of the electron transport chain, they are no longer high-energy electrons, they're now just plain old ordinary electrons, and we have to get rid of them. The way we get rid of the used-up electrons is to add them to hydrogen ions, which makes hydrogen atoms, and then we add the hydrogen atoms to an oxygen atom to make a water molecule. These are the water molecules that we saw on the right-hand side of the balanced chemical equation in the level 1 video. And, by the way, this is why you die without oxygen. Without oxygen, you can't carry away the used-up electrons at the end of the electron transport chain, and the electron transport chain backs up. When the electron transport chain backs up, the citric acid cycle backs up. And when the citric acid cycle backs up, you might be able to keep glycolysis going if you can do anaerobic fermentation and turn pyruvate into lactate, or, your other choice, you just die. But sometimes when you're running away from a tiger, your skeletal muscle cells can do the anaerobic glycolysis and fermentation trick to at least keep a couple of ATPs being made per glucose. 6 carbon atoms came into cellular respiration in the glucose molecule, and 6 carbon dioxide molecules were thrown out. 2 in the pyruvate dehydrogenase complex, and the last 4 in the citric acid cycle. We made a whole lot of high-energy electrons, and we sent them to the electron transport chain to turn their potential energy into ATP. From each glucose molecule, glycolysis made a profit of 2 molecules of ATP and a pair of high-energy electrons on NADH plus H+. We may or may not be able to use those high-energy electrons from glycolysis, but it depends on details that we don't care about right now. The pyruvate dehydrogenase complex makes 2 pairs of high-energy electrons on NADH plus H+, for each glucose molecule, 2 pairs. The citric acid cycle makes 2 molecules of GTP, 6 pairs of high-energy electrons on NADH plus H+, and 2 pairs of high-energy electrons on FADH2. We can make a theoretical maximum of 3 ATPs per pair of high-energy electrons on NADH plus H+, and a theoretical maximum of 2 ATPs per pair of high-energy electrons on FADH2. But okay, obviously in real life, we're never going to get the theoretical maximum yield. For a conservative estimate of around 2.5 ATPs per NADH plus H+, and around 1.5 ATPs per FADH2, the electron transport chain can give us 20-odd to just a little over 30 molecules of ATP from each molecule of glucose, compared to the 2 molecules of ATP we can get when all we can do is glycolysis. So yeah, the electron transport chain is where the money is. Okay, that's enough for now. In the level 3 videos, we're going to look at each one of these pathways one at a time with, I'm afraid to say, more of the horrible, horrible details. In the meantime, thanks for watching. I'll talk to you later.